Okay, good evening, everyone. A very lovely. So today we are very interested in protein, right? So with the what are now what are probably the polymers of amino acids, right? So whatever property these amino acids having given to the protein, right? And amino acids we have already in our previous video. What are the what how they are classified as per their you can say the R as per the charge as per their metabolic. Now here I think acids are going to now always remember one amino acid combining the second amino acid, it always loses molecule of water. Please remember. So once it loses a molecule, it forms a peptide. So we can say that a peptide bond is produced when one amino acid combines with the second amino. Right? Now see the most important here is that this peptide bond is the strongest bond. We can say that this is the strongest, which means that it cannot be broken via denaturation. Always remember. So we can say broken can't be broken via denaturation. Right? So this is a very important question. Also, that name the bond cannot be broken via denaturation. So the only is that peptide. Okay. So now there are different biological. Jैसे we have discussed that dipeptide is then tripeptide, and if more than 3 polypeptide is produced, right? So if a polypeptide is produced, always remember it two terminal. It has one N terminal and it has one C terminal, right? So if we talk about any protein, basically it this type. One is N terminal here, the other is C terminal. Here, today we are going to discuss how these amino acids aligned with one another to form a protein, or what is the structural organization of the proteins. Because always remember one thing, that the proteins, yeah, anything tends to go in a stable conformation. Just as suppose if you are attending my class, you will keep on changing your position so that you can understand to the maximum. Okay, so hello. Okay, right now it's okay now. Okay, right. So, so we can say that a protein will always have a N terminal and a C terminal. Clear? Now, what we are going to discuss today is the structural organization of proteins, which is very, very important. Structural organization of proteins. Right. Now, what we are going to study in this is everything tends to go in a stable conformation. Just for example, if you are also sitting, yeah, if you are listening to my lecture, you will keep on changing your position. Some students will find it more comfortable sitting, lying on the bed, and listening to the videos. Some students would prefer sitting with a proper arrangement and listening to the lecture. So it always varies, right? Same is the case with proteins. They always tend to go in a stable conformation so that they can give their maximum productivity, right? So if we talk about the structural level of organization, there are four levels, right? Primary level, secondary level, tertiary level, and quaternary level. So these are the four levels. Now, obviously, if we talk about the primary one will be totally the basic one. Secondary, little bit more higher. Tertiary, obviously more higher. And quaternary will be the most complex one. So there is a hierarchy of the structural organization of proteins from primary moving to what secondary, tertiary, and quaternary. So what will be the number of the bonds? What will be the type of bonds? Because bonds are the ones which are going to give stability to any molecule. So if we talk about the primary structure, okay, if we talk about this, so always remember primary structure tells us about the linear sequence of the amino acids in a protein. Primary structure tells us about the linear sequence of amino acids in a protein, right? Which means that it has a straight line structure. For example, I can say that if it is linear, alanine is there, valine is there, leucine will be there, say isoleucine will be there, right? So I can say that the only bond which will exist here will be the peptide bond. Clear? Clear, beta? So always remember, primary structure depicts the linear sequence of the amino acids in a protein. Clear? Right? So can anybody tell me what will be the name of the bond here? The bond present in the primary structure will be only peptide bond. So I can say that only peptide bond will be there. Will be present. Right? Now, if we go on, we are moving towards ready. I don't know, beta. I think there was some problem with the audio thing. Now I think it is clear. Now can you hear? Now I think my sound is crystal clear, I hope. There was some technical issue in that. Is it clear now? So in primary structure, the peptide bond will be there. Clear? Then moving on after, now also it's not clear. Maybe some network issue is there, I think. I have a feeling. So whatever I am speaking, I am writing also. My screen is visible, na? So just try to correlate it with this. So in the primary structure, we can say that it is depicting the linear sequence of the amino acids in a protein. So peptide bond will be there. Now, if we move towards the secondary structure. Wait, I'll just inform. 1 second. One second, beta. This. Yeah, thanks. Ready for understanding. Baki rest they are trying to resolve. Let's see. Then comes the secondary structure. Right? Now, if we talk about secondary, obviously it will be higher than the primary structure. So, secondary structure may always remember beta, peptide bond will always be there. Okay, but along with peptide bond, some other bonds will be there. And what are those bonds? The bonds are, one is peptide bond, right? Right? One is peptide bond, and always remember along with peptide bond, hydrogen bonds are also there. Hydrogen bonds. And these hydrogen bonds are, one is intramolecular and second is intermolecular. Right? And the third bond along with peptide and hydrogen bond are disulfide bonds. Right? So I can say that the three bonds present in secondary structure are the peptide bond, the hydrogen bond, and the disulfide bonds. Right? And primary structure, if you see, primary structure has only one bond, which is peptide. Clear? And this secondary structure exists in two types. It is of two types basically. One is your alpha helix and second is beta pleated. One is alpha helix, other is beta pleated. Right? Now, if we talk, yes, very good, ready. One is alpha helix, yes. So I can say that the basic difference between primary structure and secondary structure is, primary mein there is only one bond, that is peptide bond, and in secondary structure along with peptide bond we have hydrogen bonds, we have disulfide bonds also. This alpha helix can be right-handed like this, it can be left-handed like this. Right? Different proteins are stable in this alpha helix. And if we talk about beta-pleated, always remember beta-pleated has again two types. And what are the two different types of beta-pleated? One is parallel and the other is anti-parallel. Right? Now, what are the important points in alpha helix and in beta pleated? If you talk about alpha helix, I can simply say that between the first amino acid and the seventh amino acid, there will be a intramolecular hydrogen bond like this. So I can say that alpha helix has intramolecular hydrogen bonding. And if we talk about beta-pleated, I can simply say if it is parallel or it is anti-parallel, right? It will have intermolecular hydrogen bonding like this. Similarly, it will have disulfide bonds also like this. So I can simply say that here what will happen is it will have intermolecular hydrogen bonding and intramolecular. But it exists in both sides, right-handed also, left-handed also, depending upon from where the coiling is starting. It can start from here, it can start from here, but most stable is your right-handed alpha helix. Okay, otherwise it can be left-handed also, but stable one is right-handed. Clear, ready? And intramolecular hydrogen bonding. Right? Now, the most important, and I will say disulfide bonds are also there. And you know, very interesting example of disulfide bonds. Okay, disulfide bonds, जैसे curling of hair is there, straightening of hair is there. Some, you know, girls are interested in having those curly type of hair. Some wants to get their hair straightened up. So what happens is, this is the reaction of the disulfide bonds. There is one reagent dimercaptoethanol which acts on these disulfide bonds. So I can simply say curling and straightening of hair will come in this. Straightening of hair. So this is secondary structure and alpha and beta helix. Now the most important point, which is important from examination point of view also, and it is asked so many times, that proline is never found in secondary structure, neither in alpha helix nor in beta helix. Proline never found in alpha helix and beta pleated. Very, very important. And the reason I have already told you, that whenever we look at the structure of proline, whenever we look at the structure of proline, it is having a pyrrolidone ring, and it is basically a amino acid, right? And the alpha carbon atom is not free. So since the alpha carbon atom, if it is not free, what will happen? If alpha carbon atom is not free, it cannot react with the other reagents, yeah, other amino acids. That's why it is, wherever it is present, it will destabilize the helix, right? So this is proline, and this is secondary structure. Then there are some super secondary structures also. There are some super secondary structures. What do you mean by super secondary structures? That they are beyond secondary. Right? कहते हैं ना super से भी ऊपर, like that. So they will not come under secondary, they will be beyond secondary. Why? Because obviously they will try to go in more stable form by having different other bonds. So obviously the nature of the bonds has to change in super secondary structures. And always remember there are only, yes, you can say segments of polypeptides linked to secondary structure. This can also be the definition, right? And there are only three super secondary structures. And what are they? First is your beta alpha beta, right? Second is your beta meander, right? And third is your Greek key. Yes. So beta alpha beta, beta meander, and Greek. What will be the difference between these per second structure? Okay. So if I talk about beta alpha beta, you can see that there is a mergence of beta alpha with beta, isn't it? So the structure would be like, suppose if I say this is beta, this is beta, it is connected by alpha. Getting my point? So this is how we define the stability. Beta turns and loops are also there. So many are there. Because I told you know what is the logic behind this? What is the logic behind the structural organization of proteins? Plain and simple logic is that they always tend to go in a stable conformation. So to go in that stable conformation, there will be a change in the bonds. Okay, so many are there. Reverse turns, which we are talking about, reverse turns are there, beta bends are there, they will bend. Why? Because they want to go in a stable conformation. And what is the need of going into stable conformation? Because these proteins are the basis of everything. Getting my point? Yes, yes, you better add. And this is a question which has been asked, beta. This is very important. And you know how the question is asked? Question is asked like this, all come to the category of super secondary structures except, I don't remember the year, but this question has been asked. So all belong to the category of super secondary structures except those में these three options were given. The fourth option was this reverse turns. So always remember there are only three super secondary structures: beta alpha beta, beta meander, and Greek. This you have to remember like this. Okay. Up now, beta meander, why we call them Greek key and beta meander? Because they are like that. Greek key, if we talk about, it resembles Greek pottery. So it is like this, like this, like this. Mergence of beta and alpha along with beta. Then just now Reddy also said that others are also there. Beta bends are there, reverse turns are there. They are again making it stable. Then we come over to tertiary structure. Right? Now, if we talk about the tertiary structure, always remember that tertiary structure is the most stable one. Means whenever any protein is in tertiary structure, it is considered to be the most stable one. Or we can say that whenever we talk about any enzyme, we know that all enzymes are proteins. Isn't it? So all enzymes, whenever we have to study the structure of enzymes, we have to go to the level of this tertiary structure. Yes, I am coming to that. Yes, very good, ready. So now, first of all, what are the I'll write most stable. Now, if we talk about the bonds, so if we start from the basic bond, I will start. So the first bond which is present in tertiary structure has to be peptide bond, right? Second bond along with peptide bond is hydrogen bond, right? Hydrogen bond can be intramolecular, can be intermolecular. Third type of bond, ionic bonds, disulfide bonds, right? But The bonds giving stability to tertiary structure. The bond giving stability to tertiary structure are hydrophobic bonds. Very, very important. Hydrophobic bonds giving stability. Right. अब question arises, what are these hydrophobic bonds? अब देखो, hydro means water, phobic is phobia. They are scared of water. Okay, so if they are scared of water, they will try to put themselves inside other. They are going to form some cluster of structure, like it. They will form this type of structure. Means like this. Okay, so if they are forming a clustered structure, we can say that they will be more stable, and only the portion which is, yes, very good, ready. So the portion which is exposed behaves as the active site. So I can say that the bond which provides stability to the tertiary structure are hydrophobic bonds, and hydrophobic bonds are formed from those amino acids which are aliphatic. Leucine is there, isoleucine is there, valine is there. So they will tend to cluster themselves. So whatever is projected outside, that will become the active site. So obviously, if we know the active site, we can plan the mode of action. How the enzymes are functioning. And iska example, I'll just give you one common example. Whenever we construct a house, we always keep one main door open, isn't it? The gate, from where everybody can make an access. Here also the same thing is there. So it is going to form a domain type of structure. So if I ask you, domain is the characteristic structure of what? So immediately one thing should come to your mind, that domain is a characteristic feature of only the hydrophobic bonds. Right? And what are hydrophobic bonds? Simply they are scared of water, or you can say they are the ones which give characteristic stability to tertiary structure. Then we can say that tertiary structure is best visualized. This is also very important. Best visualized by X-ray crystallography. Very, very important. जैसे normally we used to say earlier that chromosomes are best viewed in their metaphase stage, isn't it? Yes, very good, ready. Chromosomes are best viewed in their metaphase stage. Same is the case with this. Enzymes can be best viewed when they are in tertiary conformation. Very important. Enzymes can be best viewed whenever we can see them in tertiary conformation, right? Then the last one is, so this is tertiary, you can say. So I can simply say this is what is the domain. Then the last one is your quaternary conformation or quaternary structure. Now, if we talk about quaternary structure, it is the most highest form. And here what is happening, beta? Here the molecules are combining. Say one chain is like this, it combines to form a quaternary structure, and these are the polypeptide units. Right? And this is the most complex one. And if we have to write the example of quaternary structure, we can simply say the example will be, if I talk about LDH. LDH we have studied in detail, and those who are again first prof students, I told you beta LDH is a very, very important cytosolic enzyme which converts pyruvic acid to lactic acid. Okay, so LDH is a tetrameric one. Yes, myoglobin, so many are there. Tetrameric one. Then CPK is there. It is dimeric. Hemoglobin is there. So many are there. So they are what? They are all in their quaternary structure. Right? So this is the structural organization of proteins. Primary, moving towards secondary, moving towards tertiary, and moving towards quaternary in the end. And there are certain super secondary structures also. Right? So this is what is the chemistry of proteins, which is very, very important. Now, coming over to this, just now I told you that peptide bond is considered to be the most stable bond. Peptide bond cannot be broken, isn't it? So there has to be some reagent, some technique by which we can identify the sequence of the amino acids also, because this is what is our core area. It's a common example I'll give you. We normally talk about sickle cell anemia, isn't it? Sickle cell anemia, may we all know that there is a change. If we talk about sickle cell anemia, then we know that there is a change from glutamic acid and valine, isn't it? And where it shows a change at the 6th position and beta chain. How we came to know that this is the one which is made possible? Just because we are going from quaternary structure to tertiary to secondary to primary. Right? Yes, now I'll come to this, that there has to be some method by which we can actually identify that what is the sequence of the amino acids. So for the identification of N-terminal residue, because there are only two terminals, N terminal and C terminal. Yes, but it is not sequencing, I would say. Sanger sequencing is for DNA. Here, this is known as simple Sanger's method of, identification of N terminal residue. So what happens is there was one scientist, Frederick Sanger, right, who gave the structure of insulin also. So what Sanger said, that there was one reagent which is, can you tell me the reagent? Yes, let me see. Ready, what is Sanger's reagent? 2,4. Tell me. Yes. Dinitrofluorobenzene. So second or fourth पे it has dinitro group, or first position पे it has fluorine. Okay. So this is required for N-terminal residue analysis. And what happens is, say, this is your N-terminal, this is your C-terminal. Whenever we treat it with Sanger's reagent, one amino acid will come out. So suppose if we have 10 amino acids, one will come out, it will be left with only 9. Then slowly, if we keep on doing it, we can say that the sequence of amino acids is known to us. So this is Sanger's reagent. Very, very important. Sanger's reagent is required for the identification of N-terminal residue analysis. Clear? And then after that, and this Sanger was the first scientist, I should say, who gave the structure of insulin also. And he was awarded a Nobel Prize also for this. And Sanger's reagent के बाद another one more is there, Edman degradation. Edman's degradation. And what is Edman's reagent? Edman's reagent is PIR FITC, which stands for phenyl isothiocyanate. Same thing, it also is N-terminal residue analysis. Is it clear? This is very important. This you have to understand. I will just, because I am trying to make it more simple. Reverse, I didn't get you, Edmund, and also reverse. What is reverse, beta? This is basically identification through the N-terminal residue. Because just now I told you it is difficult to break the peptide bond. So peptide bond can only be broken with the help of these reagents. So Sanger's and Edmonds, basically the most frequently commonly used method is Sanger's method. Then comes this Edmond degradation. And if we talk about C-terminal के लिए there are certain enzymes जैसे carboxypeptidase, chymotrypsinogen and all. But normally we always go through identification of N-terminal residue analysis. Right? So this is all about the chemistry of the proteins and the amino acids. Then after this, then we come over to a reverse method for primary structure. Okay, yeah. But the most commonly used method, beta, it is Sanger's method only. Then we come over to protein classification. Okay. Now there are so many different ways of classifying proteins. As per their length, proteins were classified into fibrous and globular. Okay. As per their functions, they are classified into जैसे defense proteins, structural proteins, then your transport proteins, flavoproteins. But the most widely accepted classification is which says that proteins are classified into 3 classes. Right? Yes, globular is there, fibrous is there, then as per their function, and just say functional proteins, flavoproteins, but we always go through this accepted classification. Yes, which says that proteins have simple, conjugate, and derived. These are the 3 main classes of proteins. And if we talk about, yes, very good. If we talk about simple proteins, simple means they are simple, as it is clear from the word itself. And what will be the examples? The most important one is albumins, globulins, right? And along with albumin and globulins, all the simple proteins which are present in the diet which we eat. Just if I talk about glutenin, glutenin is the one which is present in wheat. If I talk about zein protein, zein is present in maize. If I talk about originin, which is present in rice and barley, and then I can say simply scleroproteins, right? And histones. They will come under simple proteins. And then if we talk about conjugate proteins, conjugate means they have a simple protein which is attached to a non-protein part which is the prosthetic group, some other protein. Okay, so इसमें example will be if histones combined with DNA, they are known as nucleoproteins. Right? Then comes chromoprotein. So many are there. Chromo means colored proteins. As hemoglobin is there. Right? Then flavoproteins. Riboflavin will come. Yes, nucleosome is, yes, very good. Nucleosome is better when DNA combines with histones and non-histones. Yes. Right. Then flavoproteins. Then the most important class is lipoproteins. And these lipoproteins you are going to study in detail in lipids also, because they are having lipid plus protein. And there we will study how much amount of lipid is present, how much amount of protein is present. Right. And you can say that along with this, I can say phosphoprotein is there. Casein. Casein is the most important protein which is present in milk. So these are the conjugate proteins, right? Then we come over to derived proteins. Derived, as it is clear from the word, they are derived from primary or this simple or conjugate. Okay. So derived proteins में I will simply, वैसे तो they are of two types, primary and secondary, but simply you can write जैसे gelatin is a derived protein from collagen. Gelatin which is present in the jelly and all which we eat, it comes from collagen. Similarly, myosin will come from myosin, like that, right? So this is the classification of proteins. This is how we classify them. And albumins and globulins, so we all know they are the very, very important plasma proteins. Okay, and they are separated also by these albumins and globulins. And globulins are separated by salting out method by ammonium sulfate. Right? You can say half saturation is there and full saturation is there. Then after this, we come over to the next segment in chemistry, the qualitative test. Very, very important. The qualitative test. Because this all will come under the chemistry of proteins. Right. Qualitative test. Now here you have to see that this is again very, very important whenever we talk about qualitative tests. Qualitative means just by looking at the quality of the reaction, we can say that it has this present, it has that present, like that. So if we talk about normal qualitative tests, so I can simply say that proteins are going to give the test first of all. Right. And similarly, proteins are formed of amino acids. So what are the different color reactions of amino acids? Right. So, beta, if we talk about the qualitative tests of proteins only, there are two tests. One is biuret test. Biuret test is given by all proteins. The only reason, the only you can say property of the amino acid to give a positive biuret test is they should have more than two peptide bonds. Yes, because bi means two, uret comes from urea. So biuret test will be given by those peptides you can say, or those amino acids which have more than two peptide bonds. Two peptide bonds, right? And that also why they have more than two peptide bonds, because here copper sulfate is there. Yes, very good, very good. It always gives a purple color because here copper is there. So copper fixes itself and it needs to have four bonds, and then only it is going to give purple color. Suppose if we have to examine, if we have to check the amount of protein in any vaccine in the serum, जैसे anti-rabies serum is there, anti-snake venom serum is there, whenever we have to quantify all the polysaccharide vaccines and the proteins, protein component is always checked by this biuret test. Just keep this thing in mind. This is very important. Okay, so this is biuret test, and second test is ninhydrin test. Ninhydrin test. Now, ninhydrin test mein you have to remember only one thing, that it shows the decarboxylation. Decarboxylation of the amino acids. Right? And always remember it is decarboxylation. So here purple color is there, it gives Roman blue color. Roman blue, or you can say little bit purplish. Roman blue or purple color. But proline gives yellow color with ninhydrin. Gives yellow color with this ninhydrin. Right? Yes, it is better Roman blue or pinkish, so very rare it is pink, but it is like bluish and purplish mix. Okay, but proline, whenever we are carrying out any test of protein which has proline in it, it will always give a yellow color, right? And after this, we come over to the color reactions of amino acids, which is very, very important. Color reactions. Yes, both will give yellow color. Yes, because main to proline is there, no? Color reactions. Now see, here you have to just remember the name of the amino acid, because this is very, very important. If I talk about xanthoproteic test, xanthoproteic test, always remember xanthoproteic test will be given by only aromatic amino acids, right? Aromatic, which will include phenylalanine. No, beta, xanthoproteic will be given by aromatic amino acids. Got it? So, phenylalanine, tyrosine, and tryptophan. But here you can simply say phenylalanine. Then, if I talk about Millen's test, Millen's test is given by specifically by tyrosine. Why tyrosine? Because it has a hydroxyl group. Yes, very good. And then comes Hopkins-Colles test. Hopkins-Colles test. This is given by tryptophan. Tryptophan, which has a indole ring. So the characteristic test for tryptophan is Hopkins-Colles test, right? Principle, nobody is going to ask, but yes, these are very, very important, which you people should know. And those students who are studying in first prof, this is a very important practical also which you people perform. So this is very important from Viva point of view also, plus practical point of view also. Then Sakaguchi test. Sakaguchi test is given by only and only arginine. Very important. Sakaguchi test is given by only arginine. And you know, arginine has a specific guanido group. And then after Sakaguchi, then we come over to lead acetate test. This lead acetate test is given by cysteine, which is a sulfur containing amino acid. Right, so these are beta Obermeyer is also there, but this is basically when we are discussing the inborn errors of metabolism. When we discuss about Hartnupp's disease, no, there it is detected by this Obermeyer test. These are simply color reaction tests. That is an abnormality basically, no, Hartnupp's disease, then it will give a Obermeyer test. You are right, but here we are just in general talking about the basic tests. Okay, so these are the color reactions. Right, so here we finish up with the chemistry of the proteins. So far we have discussed about the properties, about the chemistry of the amino acids and of the proteins. Then after this we will be discussing the metabolism of amino acids and proteins in the next session of the class. So I hope till here it is clear to all of you. If there is any confusion, you can ask me. You can ask me now. And if it is crystal clear, we can, I mean, like go for the next session tomorrow. Everything, whatever we have discussed today, is it clear to all of you? Small doubt, yes, ready, you can ask me. What is the doubt? Whatever is the doubt, you can ask me. Is it lead acetate or lead sulfide? But it is lead acetate test. Basically sulfide. Why? Because lead sulfide precipitates are produced. No, black color precipitates are produced. The name of the test is lead acetate, but it forms lead sulfide. So black precipitates of lead sulfide will be there. Because cysteine is having this SH group, no? That's why. Is it clear? Cyanide nitroprusside test, beta, it is again for an inborn error of metabolism when we are studying homocystinuria. If a patient is suffering from homocystinuria, then we will carry out the cyanide nitroprusside test. Okay, this is in general, I am talking about the color reactions of amino acids, that's it. When we will discuss the inborn errors, then we are going to discuss these tests. Okay, so shall we wind up the session? Anyone, anybody else who has a doubt, you can ask me. Okay, my pleasure. So shall we wind up the session? Right. Okay, so see you guys in the next session. Till then, take care. Thank you so much. Right.